Welcome to Neuroscience. I'm going to begin our neuroscience review with a clinical evaluation of the components of the central nervous system first and how they develop. First off, on the next page, we can subdivide the nervous system into its two basic components. The peripheral nervous system contains the spinal and the cranial nerves and the neuronal processes that make them up. The central nervous system basically consists of the brain and the spinal cord, elements that are formed, as we'll see, from the neural tube. Taking it together, we could say that the entire nervous system is either a derivative of the neural tube, if it's the CNS, or neurons that make up fibers in nerves are formed mainly, but not exclusively, from neural crest cells. For example, as we list at the bottom of the page, skeletal motor neurons and axons of preganglionic autonomic neurons are derived from the neural tube and grow out from it, meaning their cell bodies are still contained within it. Neural crest cell derivative neurons that will be found in nerves will be postganglionic autonomic neurons and primary sensory neurons. So simply put, neurons that are found in nerves are a combination of neural tube cells and neural crest cells. Next point that we need to make is, what are the two glial cells that form myelin for either axons in the peripheral nervous system in nerves or make myelin for a tract fasciculi lemniscus or funiculi, bundles of axons inside the CNS? And the difference is significant. Schwann cells are neural crest cell derived glial cells that make myelin for any axon in the peripheral nervous system, regardless of its embryonic origin. Oligodendrocytes make all myelin for any axon contained within the CNS, any tracts or fasciculi, including, as we will see, all of the myelinated axons of the optic nerve. The other difference is Schwann cells can only myelinate a single segment of a single axon, whereas oligodendrocytes can myelinate many segments of many adjacent segments of individual axons inside the CNS. Let's now briefly look at disorders of demyelination, and they include a central demyelinating disease, which is multiple sclerosis. This affects myelinated axons inside the CNS exclusively and also includes the myelinated axons frequently of the optic nerve and the myelinated axons of the medial longitudinal fasciculus. These patients are likely to present with altered vision, internuclear ophthalmoplegia when you affect the axons of the MLF, and they present with motor and sensory deficits that tend to come and go. This is a chronic inflammation of axons, presenting usually with increased oligoclonal banding in the CSF, and it's usually treated with high-dose steroids. Other demyelinating diseases include metachromatic leukodystrophy. This is a lysosomal storage disease where there is a deficiency of the aryl sulfatase A enzyme, and it affects myelin in both the central and the peripheral nervous system. Patients that develop progressive multifocal leukoencephalopathy, or PML, are immunocompromised patients, often characteristic of aged patients, and these present with demyelination involving limb weakness and cortical problems. Finally, when there is a progressive bilateral demyelination, typically in the rostral and basal pons, patients might present with what is known as the locked-in syndrome. This is a patient that has bilateral demyelination of both the corticospinal and the corticobulbar tracts, resulting in spastic quadriplegia and also a total loss of all cranial nerve function except for cranial nerves 3 and 4. Lastly, Guillain-Barré syndrome is a classic demyelinating disease of the peripheral nervous system only. It typically presents in most patients that have had a recent history of a respiratory or GI illness. Motor signs and symptoms are always present, usually bilaterally in the limbs. These patients also may present with autonomic dysfunction, some cranial nerve involvement, particularly medullary cranial nerves. Sensory loss is highly variable. The sensory loss can be mild or severe and present with altered sensation of touch, vibration, and pressure, including altered sensation to pain. And classically, because of the motor neuron involvement, these patients have suppressed or absent muscle stretch reflexes. The next topic we wanted to cover is the process of neurulation. and the events involved in neurulation are summarized on this slide. Note first off that neurulation begins in week 3, immediately after gastrulation. The time frame is important because even though all nervous system components are derived from a derivative of ectoderm, one of the 3 germ layers that's established at gastrulation, the notochord must be present in the developing mesoderm so it can induce the overlying ectoderm, as we will see, to form the central and the peripheral nervous system. The 3 stages of neurulation are the following. The notochord induces the overlying ectoderm initially to form a thickened portion of neural ectoderm called the neural plate. Secondly, the neural plate undergoes a subsequent invagination in the future dorsal midline and forms a pair of neural folds. Later on, those neural folds will meet and fuse in the dorsal midline to form a completely hollow neural tube. So the 3 stages of neurulation basically are the formation of a neural plate, followed by the formation of a neural groove and neural folds, finally seeing the formation of a hollow neural tube. In addition, at the same time as neural tube formation, neural crest cells also develop from the overlying neuroectoderm, but they develop outside the developing neural tube, as we'll see, near the margins of each of the neural folds. Next point down at the bottom of the page is that even at its early stages of formation, the neural tube acquires a polarity, in that the dorsal half of the neural tube will develop into what's called the alar plate. In the alar plate, we will see the development of sensory neurons. The ventral half of the neural tube will give rise to a basal plate, and in the basal plate, we will see the development of motor neurons inside the central nervous system. And the plates, as we see here, are going to be separated by an embryonic sulcus limitans. Note, as we pointed out earlier, that neural crest cells mainly will form neuronal processes and neuronal cell bodies that are associated with nerves. But being specific at the bottom of the page, they will form all primary sensory neurons in cranial and spinal nerves, and they will also give rise to all of the postganglionic autonomic neurons that are also found in cranial and spinal nerves. And finally, as we know, the peripheral nervous system, we said, consists entirely of nerves, and we're just providing the numbering scheme. There are going to be 12 pairs of cranial nerves and 31 pairs of spinal nerves, each with a combination of neural tube and neural crest cell derived neurons. Next, this page, we're going to look at a series of diagrams that illustrate the process of neurulation. And the picture on the left side of the page illustrates a view of the future dorsal midline of the developing embryo at about day 18. And we're looking down at basically what is a view of the overlying ectoderm. And what we're seeing superimposed on the diagram is the beginning of the appearance of the notochordal process that develops in the underlying mesoderm. And in the cross-section indicated by letter A over on the right, we see a trilaminar embryo. What you're seeing here is the position of the notochord in the future dorsal midline and the fact that it has now released inductive factors that have caused the overlying ectoderm to thicken first and form a neural plate out of neuroectoderm. And what you're seeing at letter A is the secondary formation of a shallow neural groove lined on either side by a pair of neural folds. In B, we can see that the neural groove gets progressively deeper and the neural folds elongate, and we also see developing at the lateral lips of each neural fold the series of where neural crest cells will also develop. Finally, in the next picture, we can see at letter C that the lips of each neural fold have migrated toward the midline and have fused to transform a shallow neural groove into a completely enclosed hollow neural tube. And once neural tube formation is complete, the neural tube sinks down and becomes completely surrounded by mesoderm, and then the site where neurulation took place is overgrown by surface ectoderm, so there's no evidence in postnatal life that neurulation was a surface ectoderm event. Secondly, you can see that the neural crest cells develop at the same time, but develop in a position that sees their location outside the developing neural tube. And down in figure D, you can see their classic dorsolateral position where they form initially adjacent to the neural tube. And this position outside the neural tube will give neural crest cells the ability not only to form classes of neurons and nerves, but they also are now in a position to migrate throughout the body and form a wide variety of non-neuronal cell types. The picture on the left side of the page illustrates a longitudinal view of neural tube closure and makes the point that the neural tube doesn't close simultaneously all at once from the rostral to the caudal end. The point that's illustrated over on the picture on the left is the middle of the neural tube actually closes first, and this occurs approximately about day 22 or day 23. Then the rostral neural pore closes second about day 25, and then the caudal neural pore closes third and last about day 27. Secondly, this similar figure also illustrates the differences in the two most common forms of open neural tube defects. They occur either when the rostral neural pore doesn't close or when the caudal neural pore doesn't close. If either end of the neural tube doesn't close, pregnancy is going to be characterized by elevated levels of alpha-fetoprotein that's detectable in amniocentesis. What distinguishes anencephaly, which is a rostral neural tube defect, from the most severe form of spina bifida, which is a caudal neural pore defect, is that a pregnancy with an anencephalic fetus will also be complicated by polyhydramnios, because if the rostral neural pore fails to close, there is no neural mechanism available for swallowing. But if a fetus has a caudal neural pore defect, amniotic fluid levels during pregnancy will be normal, since the caudal end of the spinal cord plays no role in the swallowing reflex. Next, let's look at post-neural tube closure formation of the nervous system. Over on the left side of this figure is the completely formed neural tube. And the point we want to make here is that during fetal life, the rostral end of the neural tube undergoes much greater development than does the caudal end. And what we see is that the rostral neural pore undergoes a process of cephalization, where we see that it forms initially three primary brain vesicles, known as the forebrain, the midbrain, and the hindbrain. And then from there, those three primary brain vesicles subsequently elaborate to form 5 secondary brain vesicles. And it is from those 5 secondary brain vesicles that all parts of the CNS will form except for the spinal cord. So let's now document them, and it'll give us an idea of what kind of postnatal elements of the CNS are derived from a particular secondary brain vesicle. First off, you can see that derived from the telencephalon are mainly the cerebral hemispheres, and we'll add the basal ganglia to this list when we look at a chart down below. The diencephalon gives rise to the thalamic derivatives. That's the second of the 5 secondary brain vesicles. The brainstem forms from the caudal remaining secondary vesicles, the mesencephalon, the metencephalon, and the myelencephalon. Mesencephalon forms the midbrain. The metencephalon forms the pons and the cerebellum, and the medulla is derived from the myelencephalon. The spinal cord is not derived from a secondary brain vesicle, and as such, it in postnatal life is not that much different from the newly formed neural tube. The other point that we want to make here is at the time of neural tube formation, the neural tube basically envelops a hollow neural canal. And it is from that neural canal that the ventricles and the channels that connect the ventricles persist in postnatal life. So in essence, the entire central nervous system remains hollow, and these ventricles are going to be sites where CSF is produced and circulated. Note that the telencephalon, each cerebral hemisphere, will contain a lateral ventricle, meaning that there are 2 of them. The diencephalon is going to surround a third ventricle, and the 4th ventricle is going to be situated between the cerebellum above and the pons in the upper part of the medulla below. Note that the midbrain is the only secondary brain vesicle that does not contain some or all of a part of a ventricle, and the neural canal remnant that runs through the length of the midbrain is going to be the cerebral aqueduct. Note that there is also a neural canal remnant with some clinical significance, as we'll see, that runs the length of the spinal cord in the form of the central canal. In the next chart, let's be a little more detailed about exactly what parts of the postnatal brain are developing from the 5 secondary brain vesicles. As we alluded earlier, we said that the major telencephalic remnant will be each of the cerebral hemispheres, but as noted on this chart, and we'll see this subsequently, most of the basal ganglia are subtelencephalic structures that develop at the base of each hemisphere. Diencephalon, easy to remember, the diencephalon contains 4 neural remnants that each end in the word thalamus. The thalamus, as we'll see, is a combination of motor and sensory relay nuclei, meaning that any neural system that desires to influence an area of cerebral cortex must relay first through a motor or a sensory nucleus in the thalamus. The hypothalamus, as we'll see, is a major neural center that controls autonomic function and endocrine function. The subthalamus is a basal ganglia structure. It will be linked to one of the basal ganglia pathways that are closely tied with the subtelencephalic components of the rest of the basal ganglia. And finally, the epithalamus is a fancy word for the pineal. It includes other nuclei in addition to that, but certainly the most significant epithalamic derivative is the pineal gland itself. Note that the optic nerve, all the way out to and including the cell types in the retina, is not just an outgrowth of the CNS, explaining or logically indicating that all of its axons are myelinated by oligodendrocytes. It's specifically an outgrowth of the diencephalon, meaning that when the optic nerve axons re-enter or basically branch out into the CNS, in a major way they're going to synapse with diencephalic neuronal derivatives. And then looking further, we know that the remaining 3 secondary brain vesicles form components of the brainstem. They're listed here. The mesencephalon gives rise to the midbrain, and on the next page, the metencephalon gives rise to the pons in the overlying cerebellum. Myelencephalon gives rise to the medulla. And the spinal cord, as we said, is not a secondary brain vesicle derivative. It's a direct derivative of the neural canal that does not undergo such specialization. And again, just a summary in chart form of the most common congenital malformations of the nervous system, some of which we've already discussed. Anencephaly, we know, is a failure of the rostral or the anterior neuroportic close, and it commonly, as we see, will present with any neural tube defect with elevated levels of alpha-fetoprotein, but it is unique in that it is the only congenital malformation of the CNS that will also be seen with polyhydramnios complicating the pregnancy. Now, let's be a little more detailed about the different forms of spina bifida. We indicated that spina bifida results from a lack of caudal neuropore closure, but there are 3 other forms of spina bifida in which the caudal neuropore does close and form normally, but what the fully formed spinal cord fails to do in most of the forms of spina bifida is induce the formation of a vertebral arch to grow around it. So in the 3 more common forms of spina bifida, what will be seen typically at lumbar vertebral levels will be a series of missing lumbar or sacral spinous processes. What's shown first on this page is actually the most common form of spina bifida. This is spina bifida occulta, which is not only the mildest form, it is largely asymptomatic. Because the spinal cord forms normally, it's situated in its normal position in the vertebral canal, so the only structural defect evident in this newborn will be a series of a couple of missing lumbar spinous processes. And classically, this newborn individual will show a tuft of hair situated over the missing spinous processes. We indicated this is asymptomatic in all regards because neurologic function is normal. AFP levels and amniotic fluid levels are also going to be normal during the pregnancy. The only hint that there is an evidence of spina bifida occulta, the hidden form, asymptomatic form, again, is the tuft of hair that appears over where the spinous processes are missing. The more symptomatic forms of spina bifida are shown on the next 2 slides, and these are both forms of spina bifida cystica. In this case, again, The spinal cord forms normally, as indicated on the picture, but again, it fails to induce the complete formation of a series of vertebral arches. So in both cases of spina bifida cystica, as well as with spina bifida occulta, there are a series of missing spinous processes, typical at lumbar levels. But now what's different is there is going to be a cyst-like protrusion where the spinous processes are missing, and this cyst-like protrusion forms a seal or a cyst. So there are two different forms of this. In both forms of spina bifida cystica, the cyst is lined by two layers of the meninges, the dura and the arachnoid, meaning that the fluid environment of the cyst will contain cerebrospinal fluid. So this first form is literally known as spina bifida cystica with meningoceal. And since it is a form of a body wall defect, even though the neural tube forms normally, this pregnancy is likely to show an increase in alpha-fetoprotein, even though, as we see, the spinal cord is not only formed normally, it's situated in its normal postnatal position. The more severe, but actually more common form of spina bifida cystica is shown on the next page. And this is a newborn that has spina bifida cystica with meningomyelocele. Again, the cyst has the same anatomic configuration. The cyst is lined by the dura and the arachnoid, and the fluid environment of the cyst, again, will be cerebrospinal fluid. But in this case, the caudal portion of the spinal cord, even though it forms normally, is displaced into the cyst. And this newborn is likely to have altered sensation or even lower limb weakness as a result of the stretching of lumbosacral spinal nerves. The other important tie-in is that a fetus or an infant that has a spina bifida with a meningomyelocele is often likely to also have Arnold-Chiari malformation type 2 that's commonly seen in conjunction with this condition. Finally, the most severe form of spina bifida is the true open caudal neuropore defect, and this is a fetus that develops spina bifida with myeloschisis, literally meaning split brain, where there's a total absence of caudal neuropore closure, and the neural folds that basically fail to form a neural groove and a neural tube lie open on the surface of the lower lumbar region. And certainly, like any open neural tube defect, there's going to be elevated levels of alpha-fetoprotein that are detectable during the pregnancy. Finally, we mentioned Arnold-Chiari malformation briefly. There are two types. Arnold-Chiari malformation type 1 is largely asymptomatic, and it's simply a downward displacement of part of the paravermal cerebellar hemispheres, the tonsils, through the foramen magnum. The more severe and symptomatic form of Arnold-Chiari is type 2. It is a combination of not only downward displacement of the cerebellar vermis, but also tends to produce downward herniation of the medulla of the brainstem. And this largely causes an obstructive hydrocephalus because it compresses the fourth ventricle and its exit points for cerebrospinal fluid. It's also frequently seen, as we saw earlier, with a lumbar meningomyelocele, and it also tends to cause an accumulation of cerebrospinal fluid in the central canal of cervical or thoracic spinal cord, resulting in a condition known as syringomyelia. Lastly, a couple of other defects. Dandy-Walker malformation is a process where there's basically lack of proper development of the cerebellar vermis and maybe even the splenium of the corpus callosum. And particularly when the cerebellar vermis is involved, it typically is seen with a failure of one or more of the foramina of the fourth ventricle to open, causing a form of a communicating hydrocephalus. Different forms of hydrocephalus will be covered subsequently, and certainly individuals that have either the Dandy-Walker malformation or Arnold-Chiari type 2 may present with a non-communicating hydrocephalus. Non-communicating hydrocephalus is basically an obstructive hydrocephalus, and as we'll distinguish at some point, communicating hydrocephalus is most commonly an absorption problem. But regardless, both tend to present with elevated CSF volume and or CSF pressure. Finally, in holoprosencephaly, there is an incomplete separation of the cerebral hemispheres, resulting in a single telencephalic ventricle, and this is seen in a fetus that basically has trisomy 13. Let's now summarize the embryonic origin of the derivatives of both the central and the peripheral nervous system. And this story is illustrated on a chart, it's a pretty good one, that illustrates not just the nervous system derivatives, but the tissues and organs that are derived from all of the three primary germ layers. Note, as we've said, that all of the central and the peripheral nervous system are derivatives of ectoderm, but certainly surface ectoderm gives rise to a wide variety of surface structures, including the epidermis and its derivative structures, hair and nails, the hair cells and the fluid environment of the inner ear as well as the external ear. And probably the most significant thing on this list is that the overlying oral ectoderm basically develops into Rathke's pouch, which gives rise to the anterior pituitary. And the reason we make that distinction is that the anterior and posterior pituitary are both ectoderm derivatives, but they are derived from very different components of ectoderm, as we will see. This next chart subdivides the components of ectoderm that give rise to either the central nervous system in the form of the neural tube or neurons that are found in nerves that are derived from neural crest. Again, both the neural tube and neural crest are subsets of neural ectoderm, which was induced to form by the underlying notochord. And again, we know that the neural tube basically will form all of the components of the central nervous system, brain and spinal cord, and as we've indicated, that develop subsequently out of secondary brain vesicles. But as we said, all of the axons of the optic nerve, all the way out to and including the retina, are outgrowths of the CNS, meaning that all of their axons are myelinated by oligodendrocytes, and meaning also that the optic nerve will be the only cranial or spinal nerve that's going to be affected in a central nervous system demyelinating disease such as multiple sclerosis. Note that the pineal gland is a neural tube derivative, even though it is an endocrine gland that mainly releases melatonin. It's a derivative of the epithalamus. And here is the neurohypophysis. The neurohypophysis is a direct outgrowth of the neural tube ectoderm, whereas the anterior pituitary is derived from oral ectoderm of Rathke's pouch. Astrocytes are the most common glial cell. They provide the connective tissue scaffolding of the central nervous system. And oligodendrocytes are the glial cells that make myelin for all axons inside the CNS. Neurocrest cells are obviously the other neuroectoderm derivative, and certainly they are going to form two kinds of neurons that are found in nerves. They give rise to all primary sensory neurons, regardless of sensory modality, found in either cranial or spinal nerves. And they also give rise to all postganglionic autonomic neurons that will form a component of either the sympathetic or parasympathetic subdivisions of the autonomic nervous system. And as such, since neural crest cells develop outside the neural tube, all of their neuronal cell bodies will either be found in sensory or postganglionic autonomic ganglia. Next, note that the adrenal medulla is on the neural crest cell list because the adrenal medulla is really a form of a postganglionic autonomic ganglion, particularly sympathetic, where neural crest cells have migrated into the medulla of the adrenal gland and formed it. Note also that neural crest cells form melanocytes that migrate into the basal layers of the epidermis. As indicated earlier, they form Schwann cells, which are going to myelinate all axons in the peripheral nervous system. Neural crest cells give rise to two of the layers of the meninges, the pia and the arachnoid. The dura is derived from mesoderm. And as noted above in gross anatomy, we know that virtually all connective tissue and cartilage is derived from mesoderm, except for pharyngeal arch cartilage. Pharyngeal arch cartilage forms the templates for pharyngeal arch derivative bones that form mainly the maxilla and the mandible, as well as cartilages and bones in the visceral part of the neck. Odontoplasts develop in the teeth. They form dentin associated with the teeth. Note also that neural crest cells, in addition to forming the adrenal medulla, also form parafollicular clear or calcitonin producing cells that migrate into the interior of the thyroid gland. And finally, note that neural crest cells play a significant role in the development of the heart by forming most of the aorticopulmonary septum and giving rise to the endocardial cushions, which will contribute to the formation of atrioventricular valves.